The epithelial surfaces of the body are the first defenses against infection. Our body surfaces are defended by epithelia, which provide a physical barrier between the internal milieu and the external world that contains pathogens. Epithelial cells are held together by tight junctions, which effectively form a seal against the external environment. Epithelia comprise the skin and the linings of the body's tubular structures, the gastrointestinal, respiratory, and urinogenital tracts. Infections occur only when the pathogen can colonize or cross through these barriers, and since the dry protective layers of the skin present a more formidable barrier, pathogen entry most often occurs through the internal epithelial surfaces. The importance of epithelia in protection against the infection is obvious when the barrier is breached, as in wounds and burns, when infection is a major cause of mortality and morbidity. In the absence of wounding or disruption, pathogens normally cross epithelial barriers by binding to molecules on internal epithelial surfaces, or establish an infection by adhering to and colonizing these surfaces. This specific attachment allows the pathogen to infect the epithelial cell or to damage it so that the epithelium can be crossed, or, in the case of colonizing pathogens, to avoid being dislodged by the flow of air or fluid across the epithelial surface. The internal epithelia are known as mucosal epithelia because they secrete a viscous fluid called mucus, which contains many glycoproteins called mucins. Microorganisms coated in mucus may be prevented from adhering to the epithelium, and in mucosal epithelia, such as that of the respiratory tract, microorganisms can be expelled in the flow of mucus driven by the beating of epithelial cilia. The efficacy of mucus flow in clearing infection is illustrated by people with defective mucus secretion or inhibition of ciliary movement. They frequently develop lung infections caused by bacteria that colonize the epithelial surface. In the gut, peristalsis is an important mechanism for keeping both food and infectious agents moving through. Failure of peristalsis is typically accompanied by overgrowth of bacteria within the intestinal lumen. Our surface epithelia are more than mere physical barriers to infection. They also produce chemical substances that are microbicidal or inhibit microbial growth. For example, the antibacterial enzyme lysozyme is secreted in tears and saliva. The acid pH of the stomach and the digestive enzymes of the upper gastrointestinal tract create a substantial chemical barrier to infection. Further down the intestinal tract, antibacterial and antifungal peptides called cryptidins or alpha defensins are made by Paneth cells, which are resident in the base of the crypts in the small intestine beneath the epithelial stem cells. Related antimicrobial peptides, the beta defensins, are made by other epithelia, primarily in the skin and the respiratory tract. Such antimicrobial peptides play a role in the immune defense of many organisms, including insects. They are cationic peptides that are thought to kill bacteria by damaging the bacterial cell membrane. Another type of antimicrobial protein is secreted into the fluid that bathes the epithelial surfaces of the lung. This fluid contains two proteins, surfactant proteins A and D, that bind to and coat the surfaces of pathogens so that they are more easily phagocytized by macrophages that have left the subepithelial tissues to enter the alveoli of the lung. Coating of the particle with proteins that facilitate its phagocytosis is known as opsonization. And that's the epithelial surfaces of the body are the first defenses against infection. Have a great day.